This lecture has already been given in real time on April 16th, but I forgot to record it. This is a duplicate of the presentation. It covers the small intestine and the two basic parts. One part, the part deals with the structure of the small intestine, the histology, and the second part deals with the chemical digestion processes that happen. This picture that you're looking at that says in the caption, villi lining the lumen of the intestine, finger-like projections that the lumen side of the small intestine. One is a villus, it's the surface area, and more absorption of the small intestine into the bloodstream. The details, anyway, are listed in study question 13 and 15. Before we start, let's just kind of talk about an overview of what the small intestine. It's the location where most digestion takes place, chemically anyway. The stomach mechanically and the mouth mechanically, that's where digestion takes place, mechanical digestion. But the other thing that happens in the small intestine that's vital is our nutrients are absorbed. And so absorption of nutrients from the lumen of the small intestine goes across the wall, and then in the submucosa there's blood vessels that take up those nutrients. The other things that are included in these two processes that the small intestine is involved with include secreting intestinal juice and moving the feces or the waste products after digestion along the length of the small intestine to the large intestine. So start with the gross anatomy, and then we'll do the histology. The small intestine, because of its diameter, is as large as 7 centimeters. It extends the pyloric sphincter, where the duodenum is the first region, jejunum is the second region, and the ileum is the last. The shortest of these regions is the duodenum, which is approximately to where that bulge is located. So about right here. Come on. About to there. So it's only about 25 centimeters. And I bring that up because the most chemical and absorption take place in the duodenum. Now we'll talk about the histology. Now it does change a little bit all the way from the duodenum down to the ileum, but this is the general scheme. So there's 4 layers, just like any part of the digestive tract, and the innermost layer is the mucosa, and then the submucosa is underneath it, and then there's a muscular layer called the muscularis externa off the serosa, which covers the outside of the small intestine. The mucosa is lined with simple columnar epithelium, and you can see the villi. There's many different cell types in this epithelium, and they secrete different substances, which we'll look at next on the next slide. Submucosa varies from duodenum to ileum. There will always be blood vessels in there and lymphatic vessels, but the submucosa in the duodenum has these Brunner's glands, which I've circled one in yellow, and the Brunner's glands secrete mucus, which is alkaline, or it has a high pH. In the ileum there's Peyer's patches, and those protect foodborne protect us from foodborne infection. The muscularis externa only contains 2 layers of smooth muscle, which is typical for the entire digestive tract, except for the stomach. The stomach had 3 layers of smooth muscle. Here's the variations that occur down the length of the small intestine in both the mucosa and the submucosa. So in the duodenum you can see these nice Brewer's glands that secrete mucus. The jejunum, you don't see an extensive submucosa at all. Most of the submucosa extends into the villi. In the ileum you have submucosa again, but in the submucosa rather than Brewer's gland, you have Peyer's patches. And that makes the most sense because by this time the material in the lumen has probably already been neutralized, and there's been enough mucus and alkaline solution neutralizing the acidic chyme. So let's just look at the mucosa for a minute, because a lot happens in the mucosa, the layer that's exposed to nutrients. There are 3 different features. These include the circular folds, the villi, and the microvilli. So the circular folds you can see grossly if you were to cut open a small intestine. The circular folds really are made by both mucosa and submucosa, and they're just kind of pinchings or foldings of excess tissue. They're somewhat rigid, mixing of the chyme as it progresses through the duodenum. Really prevalent in the duodenum. It's also prevalent throughout, but mostly in the duodenum. And what this mixing does, besides mix with intestinal juice, it also mixes with the pancreatic juice that's been released into the small intestine and bile. Also, the materials that are exposed to the surface are refreshed as they churn, as they spiral. Increase the surface area for absorption primarily, but also for secretion. The microvilli are what is found on an individual epithelial cell lining one of the villi. So microvilli are extensions of the plasma membrane, just foldings of phospholipid bilayer like that. And again, that's to increase the surface area. But there's another interesting thing about these microvilli, and that is that they contain enzymes. So there are proteins, enzymes, embedded in the cell membrane at these microvilli, and when a nutrient bumps up against the enzyme will break it. There's 2 other features of the mucosa that are important, crypts and tight junctions. So crypts are a little bit like gastric pits in how they appear. Notice there's this hole here in the lumen. And if we go back to the very first slide, if I can get there, what you're looking at here is the entry to a crypt. And the crypt is important for the making of intestinal juice, and it also has some cells that are important for fighting off foodborne infections. There are these little yellow cells here. They're called, and we'll talk about them again. Between the cells, the epithelial cells of the mucosa, we have tight junctions that prevent nutrients from seeping in between cells to get to the bloodstream. We don't want that, particularly for lipids. We want them to go through the cell, be processed before they hit the bloodstream. So the tight junctions prevent nutrients from moving between cells. They also prevent any foodborne bacteria from getting across the wall of the small intestine. This next slide is just for fun, kind of. This is a villus that looks to me like it was imaged through scanning microscopy, although where I got it they didn't say that, but that's what it looks like because it's kind of fractured. The surface is fractured. And what you can see are some individual goblet cells that becomes part of the intestinal juice. So now it's time to look at the cells in the mucosa, and there's one that's only present in crypts, so I kind of separated it like this. All of the first three cells are found throughout the mucosa. So there's enterocytes, goblet cells, and enteroendocrine cells. The enterocytes are the ones that have the microvilli, and they have those membrane-bound enzymes, and they're important for absorption. Goblet cells secrete, and the enteroendocrine cells secrete two hormones. That's why the name endocrine is in them. And they're predominant, these enteroendocrine cells, in the duodenum. I should mention that the enterocytes do release a little bit of liquid, secrete some water that becomes part of the intestinal juice. Now the only cell that's different in the crypt is a Paneth cell, and you can see it at the base here. And the Paneth cell is a protective cell. You could consider it part of resistance to infection because what they secrete, these cells, defensins and lysozyme, which will break down bacteria. So intestinal juice is made up of water, mucus, and those defensins and lysozyme. There are no enzymes present in intestinal juice. So the small intestine does not secrete enzymes to float around in the lumen. So the pH is alkaline, between 7.4 and 7.8, due to the goblet cells as well as the Brunner's glands making mucus that contains bicarbonate ions, that's a base. The functions, of course, of this juice are mainly to neutralize the acidity of chyme and to fight foodborne infection, not to do any digestion. Let's look at the submucosa just a little bit before we talk about chemical digestion. So the submucosa is blood vessels. I guess my microphone stopped working and I'm not sure where it stopped, so I'm going to go with these intestinal cells one more time. The enterocytes have membrane-bound enzymes that will break substances down. Goblet cells secrete mucus, and enteroendocrine cells secrete hormones. The only other cell type are Paneth cells, and they secrete defensins and lysozyme to protect us from foodborne bacteria. So intestinal juice is made up of water, mucus, and these defensins and lysozyme. Notice that the mucus is alkaline because it contains bicarbonate, which is a base. And the mucus is produced by goblet cells as well as the Brunner's glands. The intestinal juice has no enzymes at all. There's no enzymes floating around in the lumen of the intestine. So intestinal juice is alkaline, and its functions are primarily to neutralize the acidity of chyme and to fight foodborne infection. I'm sorry if this is a repeat. I don't stopped working. And then the next thing we want to look at is the submucosa in the small intestine. Notice down here in the submucosa have blood capillaries, as well as lymphatic capillaries, and of Peyer's patches in certain regions and Brunner's glands in certain regions of the small intestine. Now, the importance of discussing these vessels in the submucosa has to do with absorption, which will nutrients are absorbed from the lumen across the epithelial lining and into the submucosa, and proteins and carbohydrates that have been digested or broken down will enter the blood. Any kind of lipid that's been broken down enters the lacteals. Now I think we're ready for chemical digestion in the small intestine. So, Every type of nutrient is digested in the small intestine. And remember, our categories of organic nutrients include carbohydrates, proteins, lipids, nucleic acids. Nucleic acids are an energy source for us, or nutrient source, so I'm not going to consume those too much, but carbohydrates, to start with, are broken down into monosaccharides. Those are the smallest monomers. Proteins are broken down into amino acids, again, the smallest form of a protein. And lipids and triglycerides are broken down into fatty acids and glycerol, which is a carbohydrate, as well as a monoglyceride. And I want to explain what a monoglyceride is. Remember that a lipid has a glycerol backbone and then 3 fatty acid chains. If you take away 2 of those fatty acid chains, now it's a monoglyceride. Okay, just so you know what it means. So all of these carbohydrates, proteins, and lipids have already been partially digested by the time the entire quantity of them reaches the small intestine. So 30 to 40% of carbohydrates are broken down due to salivary amylase. So the food bolus continues down to the stomach. 60 to 70% of carbohydrates will be broken down in the small intestine, but a lot more protein and a lot more lipids by percentage. So proteins, remember, are partially broken down in the stomach due to the presence of pepsin. And you might remember that lipids would be broken down by lipase, of which we have some in the mouth, lingual lipase, and gastric lipase in the stomach. Now, what are the sources of the enzymes that do this chemical digestion in the small intestine? If it's not intestinal juice, then where are the enzymes? Well, remember that enterocytes have microvilli with enzymes on the membrane. Those will break down all of our different types of nutrients in a little bit. But a lot of the chemical digestion that takes place in the small intestine is really performed by enzymes present in the pancreatic juice. And bile will help with lipid digestion, and that's made by the liver but then released into the small intestine. So first, let's talk about these enterocytes and the enzymes present there. So enterocytes contain these enzymes that we refer to as brush border enzymes. They are membrane-bound enzymes, and that means then that a nutrient must up against it for any chemical digestion to take place. The nutrients have to be churned or spiraled and moved to make direct contact with the lining of the small intestine. Simple molecules are broken down into individual monomers. You don't have to get too worried about the details. Here I included nucleic acids, although I won't test you on it. They are also broken down into their subunits. Okay? I drew a picture of what happens when the brush border enzymes break down a nutrient, and the example that I've drawn here is that I'm going to pretend that the triangle is a brush border enzyme called maltase, that when it makes contact with maltose, a sugar, a disaccharide, it breaks it down into two individual monosaccharides, two glucose molecules. And you can see that up here in the picture. So this is our maltose, and it just bumps up against the enzyme and produces enzyme. Here's a list of all the brush border enzymes that line the small intestine. For carbohydrates, we have lactase, maltase. For proteins, we have enteropeptidase, aminopeptidase, carboxypeptidase. There's some for nucleic acids, but don't worry about those. There aren't any for lipids. Okay, so there are no brush border enzymes that will break down lipids at all. Of these, I don't necessarily think you need to memorize them all, but one that is going to become important is enteropeptidase, because it's going to trigger pancreatic enzymes. So it's involved with protein digestion. What's important, I think, for you to remember, brush border enzymes can break down carbs and proteins. Because there's lactase listed here, I thought we should spend a couple of minutes talking about a somewhat common disorder called lactose intolerance. People that are lactose intolerant lack that brush border enzyme called lactase. And so lactose remains undigested in the lumen, and that causes a problem osmotically. So you have kind of this bigger solute than should exist drawing water into the lumen. And therefore, if water stays in the lumen, you often get kind of loose stool, diarrhea. However, there's another way that lactose can be broken down, and that is by bacteria. But when bacteria break down lactose, they produce a lot of gas, and that gas is uncomfortable. It causes bloating and cramping, not to mention what we know of as flatulence. Okay, before we look at specific mechanisms of digesting these carbs and proteins and lipids, we need to talk about the role of bile and pancreatic juice. So I'm going to start with bile, and bile is made by the liver. So cells in the liver are called hepatocytes, and they make this bile and release the bile into hepatic ducts, which you can see on this picture. The bile then travels from the hepatic duct to the common bile duct, which enters the small intestine at a little hole here, I guess we'll call it. There is a muscle that surrounds that entry point called the hepatopancreatic sphincter, and we'll talk about that in a second. First, the function of bile is to neutralize chyme. So as you can imagine, there's going to be bicarbonate in bile. And what the bile does, rather than true chemical digestion, is it emulsifies lipids. One thing that's interesting about bile production and making it available to the small intestine is that the liver continually makes bile. So the liver's up here, and the bile goes directly down to this entry point with the where it meets the small intestine. The common bile meets the the common bile duct meets the small intestine. However, if bile production is extensive, back up into the cystic duct and then be stored in the gallbladder. And here the gallbladder can withdraw water. It concentrates the bile. That's why you sometimes get gallstones, and we'll talk about that later. So this location, right, the hepatopancreatic sphincter. And it's normally closed, but when you eat lipids, it relaxes and the bile goes into the small intestine. So the components of bile include some salts, which are basically ions, and they help with the breakdown of lipids. And then phospholipids, which help us absorb lipids. And then bicarbonate, which neutralizes chyme. True chemical digestion. No true chemical digestion functions. It's more about breaking up fats grossly. There's also some cholesterol there, and there's some triglycerides in the bile. In fact, bile is the one way that the liver can rid the body of excess cholesterol and excess triglycerides. And then, of course, when we talked about the circulatory system way back when we were doing blood, to make bilirubin, and that's present in the bile as well. Here's how bile salts and phospholipids work to help us break down fats. The process is called emulsification, so you're emulsifying fats. So if the small intestine receives a big fat globule, the bile salts, because they're ions, can kind of break that up into smaller globules, and they coat the globules. The bile salts continue to break down, and also the fats kind of being moved along and broken up mechanically. The bile salts continue to coat and decrease the size of the fat droplet, and then phospholipids coat the outside, and these are then called micelles. So micelles are fat droplets coated by phospholipids, and I've written all that down right here. So the reason we want fat globules to be smaller is that it increases the surface area for pancreatic lipases to get at the fats and break them down. And bile salts do that too. So what happens for absorption is these are what actually gets absorbed across the wall of the small intestine. Since we're talking about bile, let's look at another disorder, and they form in the gallbladder, and they're composed of crystals, mainly cholesterol crystals. Cholelithiasis is the formation of gallstones, and cholelithotripsy could break them down. They can do that with sound waves. But let's find out how they are formed first. Usually they occur due to excessive cholesterol. They can also occur due to cholesterol sitting in the gallbladder for a long time, and excess water is reabsorbed. Too much water is reabsorbed. So now they're kind of, I wouldn't call them hard, but they're these sticky solutes that block ducts. Once the gallbladder does contract and try to eject bile, these gallstones get stuck in the ducts, and they can block the ducts at different points. Here's a picture that shows blockage of the common bile duct superiorly. And here's another picture, or another one example, of a gallstone that made it all the way down to the hepatopancreatic sphincter. This would impair pancreatic juice from getting into the small intestine if it wasn't for the presence of this accessory duct, which you can kind of see up here. But ultimately interfere with lipid digestion because the bile can't get into the small intestine. This is particularly important because we need the lipids to be broken down and absorbed, so we can absorb vitamins with the lipids. Another complication of gallstones is that we can't get rid of bilirubin, and so the skin and the whites of the eye become yellow, and we call that condition jaundice. Now we're going to look at the role of the pancreas in chemical digestion of the small intestine. So remember when we looked at histology of the pancreas and we did this in the endocrine system, we learned that there are these groups of cells called islets of Langerhans. This is where insulin and glucagon are made. And that's endocrine. In now are all these darker purple cells. Those are exocrine. And they are the ones that are going to make pancreatic juice that contain enzymes. The pancreatic juice will enter a pancreatic duct and hepatopancreatic sphincter to enter the small intestine. If that sphincter is closed, the juice has to wait until it opens. There is an alternate route, however, and this is called the accessory pancreatic duct. So we always have a little bit of pancreatic juice entering our. The components of pancreatic juice include bicarbonate, so that helps neutralize chyme even more. Enzymes, pancreatic amylase will break down carb. Pancreatic lipase will break down lipids. Don't worry about the nucleic acid ones. And some other enzymes that aren't really activated yet, so we call them zymogens. Trypsinogen, chymotrypsinogen, and procarboxypeptidase all break down proteins. These have to be active once they get into the small intestine. And that's going to happen by a brush border enzyme. So we'll look at that next. So this is a picture that shows that pancreatic juice, including all the enzymes, has entered the small intestine. That would include the zymogen trypsinogen, the zymogen chymotrypsinogen, and the zymogen procarboxypeptidase. Now the trypsinogen has to bump up against a brush border enzyme, and the one that will break it down usually is enteropeptidase. So trypsinogen becomes trypsin, and now it can digest proteins. Trypsin digests proteins, so while it's floating in the intestine, it comes in contact with the pancreatic zymogens, trypsinogen and procarboxypeptidase, and it can convert them into the active forms, chymotrypsin and carboxypeptidase. In addition, there's positive feedback because once trypsin has been activated, it can also activate itself, if you will, by interacting with trypsinogen, and that will then become more trypsin. So we've talked a lot about enzymes that function in the small intestine, a lot of chemical digestion. And I thought this summary slide might help you understand it a little bit better or group it in your mind. The first thing to think about is what are the nutrients that are broken down in the small intestine? All four big categories: carbs, proteins, nucleic acids, and lipids. Now there are brush border break down three of these categories: carbs, proteins, and nucleic acids. I told you didn't have to really know the names of these brush border enzymes except for Enterocyte, because as we have just seen, it's involved with activating a zymogen. There are other enzymes present in the small intestine though, and they come from the pancreas. So the pancreas is involved with carb digestion, protein digestion, and nucleic acid digestion. Please don't memorize those names of the pancreatic enzymes that break down nucleic acids, but you should know the name pancreatic amylase, and you need to know the names of these pancreatic enzymes that break down protein. Finally, for lipid digestion, we do have pancreatic lipase. So the pancreatic juice will break down all four major categories. It will help emulsify the lipids so that pancreatic lipase. I hope you find that kind of summary helpful. The last thing we need to talk about is the regulation of pancreatic juice release and bile release, because the small intestine can control to some degree how much bile it receives and how much pancreatic juice it receives, as well as how or what the pH of the juice and the bile is. So I need to back up here and talk about how it's hormonal control. So the duodenum right here has cells, enteroendocrine cells, that make two hormones, cholecystokinin and secretin. Both of those hormones are going to affect the liver. So let's talk about cholecystokinin first. Cholecystokinin actually has three targets. It's not the liver per se. It's the gallbladder, the hepatopancreatic sphincter, and the pancreas. So that's what cholecystokinin can affect. This is gallbladder contraction, so that bile gets pushed down the common bile duct. And then it also causes hepatopancreatic sphincter relaxation, so that the bile can actually get into the small intestine. Lastly, the hormone causes cells, exocrine cells in the pancreas, to make more enzymes. So we'll have pancreatic juice and bile enter the small intestine. Now, what causes the duodenum to make this cholecystokinin? The presence of lipid in the chyme. It signals the need for bile, and it signals the need for pancreatic lipase. Of course, all the enzymes are made by the pancreas. Secretin only has two pancreas, not the gallbladder, because it has a different. Its role is to change the pH of the bile and the pancreatic juice. So secretin will signal the cells in the liver to put more sodium bicarbonate, a base, into the bile, and it will signal the pancreas to put more sodium bicarbonate into the pancreatic juice. So if the chyme is, cells in the duodenum will respond to that acidity and make secretin. So that's what you missed on Thursday, April 16th, and I will post this shortly.